Dear students, today we will learn about cell cycle or cell division. Let's start this with a very simple concept that what is cell cycle? Cell cycle basically is a concept related with how a cell passes through different phases to divide into two daughter cells. So if I repeat, that what is cell cycle? Cell cycle is consisting of all those events and phases through which a cell passes and eventually divide into two cells. Normally the cell has two parts. The first part is called as interphase and the second part is called as M phase or mitosis phase. If you see on your screen, the cell is further divided into certain specific portions or phases which are named as G1 phase, S phase and G2 phase. And every phase perform a specific function. For example, G1 will play a specific role and S phase will also play another role which is different from the G1 phase, and same is the case with the G2 and M phase. So every phase perform a specific function. So now we will look towards the functions which are happening in the G1 phase. So in G1 phase, the cell prepare the proteins or enzymes that are necessary for the replication of DNA. So all these things will be happening in the G1 phase. In S phase, actual replication occurs. Okay, and in the G2 phase, again the cell will make some kind of proteins that further guide the cell to move towards the next step. So all these steps are necessary for a proper functioning or proper cell cycle division. And in whole process, there are certain checkpoints which guide the cell throughout the cell cycle. And now we will look forward towards those checkpoints. The first checkpoint is between G1 and S phase. The second checkpoint is between S and G2 phase, and the final checkpoint is in the M phase. So as I talked earlier, that every checkpoint is performing a specific function. So let's have a look on the function of every checkpoint. This first checkpoint, in which the function of this checkpoint is to check the ratio of cytoplasm to nucleus ratio. If there is appropriate cytoplasmic to nucleus ratio, then it will allow cell to move from G1 to S phase. If there is no suitable cytoplasmic to nucleus ratio, then it will not guide cell to go towards the next phase. So let's have a look on towards the function of the second checkpoint. The second checkpoint checks whether there is a proper replication occurred or not. If there is a proper application, then it will allow cell to move forward. Otherwise, it will halt the process. Halt mean to stop for a while and repair. Okay. The function of second checkpoint is to check whether there is a proper application occurs or not. So now let's uh, have a look on the function of the third checkpoint. The third checkpoint checks the proper alignment of microtubule assembly. So these are the functions which are performing by these checkpoints. So cell cycle means all the events which are occurring in a cell when it is going to divide from one cell into two cells. Cells have to basically make more cytoplasm, or you can say more organelles, and especially DNA should be replicated and equally divided into two daughter cells. Now, let's have a look at the first G1 phase, in which the cell is getting ready for the synthesis of DNA. So it is called as pre-synthetic phase. And then DNA replication starts in the S phase. After this, The cell enters into the preparation that how replicated DNA should be divided for two cells. For example, here it will make the proteins that are related with spindle formation. Once those proteins which are required for the division of cells, they are made, and then cell will start separating its chromatids and go through the process which will make two daughter nuclei and eventually two daughter cells. That is mitosis, and mitosis ends up here. But in some tissues or cells, uh, they might go again into proliferation, and in other tissue cells may exit out of the cell cycle and start resting, and then they can come into cell again if there is appropriate stimulus for the proliferation. Now there are some tissues in our body that are continuously multiplying. It means as soon as the cell completes its mitosis, it gets entered into the G1 phase, and it does not enter into the G0 phase, and G1 is again started doing its function. So there are some tissues or cells in our body which are continuously dividing. For example, skin cells, they are multiplying all the time. Another example is gastrointestinal cells. These type of cells are continuously dividing because the skin is growing every time, and same is happening in case of gastrointestinal cells. These cells, these cells always remain in working phase, in working position, just like from G1 to S phase, and then G2 phase, then again go into the G1 phase. So these type of cells are called as labile cells. So these cells which are multiplying all the time in the tissues, and these cells, once they complete mitosis, they do not enter in G0 phase. Once they complete one division, then they go for the next. So they are called labile cells.
Okay, there are some cells which remain in G0 phase, and these genes are doing housekeeping functions. But under certain stimulus or under certain stimuli, they can be forced to go back into the cell cycle. The example of this type of cells are liver, liver cells. The liver cells stay normally in G0 phase, but if they get some cut under the presence of some stimuli, then it will again go towards the G1 phase of the cell cycle. Cells in the kidney is another example of this type of cells. So overall, these cells are known as stable cells. Then there are some other tissues where cells are, normally stay in the permanent phase, and these are called permanent cells. Permanent cells means that in those tissues, once the cell divided them, they are going to G0 phase, but they cannot re-enter into the G1 phase because in some tissues, some cells or most of the cells, once they have multiplied and gone into G0 phase, they cannot re-enter into the cell cycle again. So the examples of these type of cells or tissues are neuron cells, or you can say the cells of eye. Once they went into the G0 phase, they will never ever again, become the part of G1 phase or other phase of the cycle. Okay, now let's talk briefly about mitosis. Mitosis is basically the division of nuclear material. During the process of mitosis, the replicated or duplicated genetic material which is coming from the S phase in one nucleus, it should be divided for two daughter nuclei. And the process in which cytoplasm and organelles is also dividing into two, and cell membrane started separating into two cytoplasmic and its organelles into two cells, is it is called as cytokinesis. Okay, now have a look over the phases of mitosis. But before going towards the phases of mitosis, let's have a rewind. I want to have a rewind between mitosis and interphase. And remember, do not mix the mitosis with interphase. You know, interphase is basically when the cell is planning to divide, or you can say getting ready for division. That include G1 phase, it is also known as presynthetic phase, then synthetic phase, and then G2 phase, or it is also known as pre-mitotic phase. So now there are some basic changes which are going or which will take place in the prophase. But before going to prophase, let's have a look over this stage of the cell, which is coming from the pre-mitotic, or you can say G2 phase. It is having a condensed chromatin, and nuclear membrane will start disappearing a little, and the chromosomes are becoming visible, and you can see the centrioles will be at the pole. So this is the prophase. After this prophase, the microtubules will be established, and they will become attached to the kinetochore, as you are seeing in this slide. So this stage is known as metaphase. And after metaphase, when you able to see structure like this, in which the two chromatids become apart, then it is called as anaphase. So we have a clear picture about what is happening in the prophase, what is happening in the metaphase, and now you are seeing that what is happening in the anaphase. And now let's move towards the very last part. At the very end, there is a telophase in which the cell will start dividing its cytoplasm, and the cell is planning to become to two daughter cells. So this is a whole overall, overview and some detail related to the cell division and mitosis. And in the next lecture, we will be talking about some stages in the and phases of the meiosis.
So now we will discuss about meiosis. But before going to the meiosis in detail, let's have a brief introduction about gametogenesis. Gametogenesis is the process by which the early germ cells undergo the process of meiosis and cytodifferentiation and eventually convert into mature gametes. Okay, meiosis is a special type of cell division which takes place in two stages, such as meiosis 1 and meiosis 2. And during meiosis, chromosome number is reduced to half in final daughter cell. So meiosis is also called as reduction division. So now what actually is gametogenesis? Gametogenesis is a process in which the germ cells convert into mature gametes. For example, in female, the gametogenesis is called as oogenesis, and in male, the gametogenesis process is called as spermatogenesis. Okay, in female, during gametogenesis, or you can say oogenesis, the germ cell passes through a process of meiosis, and then they undergo the process of cytodifferentiation and then maturation, and eventually they produce mature gamete, which is called as mature ovum. So what happens in case of males? Uh, in male, the process of gametogenesis, as we talked earlier, is called as spermatogenesis. So during spermatogenesis, again there is a process of meiosis, which again reduces the chromosome number into half in the final daughter cell, and again there is a process of cytodifferentiation, which will make the mature germ cell
in final development of mature sperm.
So now we are going to have a look at the process of meiosis in the human diploid cells. So here we have a cell prior to the meiosis in the stage of interphase. And I want to put a note in the upper left corner that notice that there are 46 strands of DNA, or you can say 46 strands of chromatin in this cell, because this is the example of human diploid cell. And if we actually just do a quick count through, and we are finding that 23 strands of DNA in black, and here the black color represents the parental DNA, the DNA that you inherit from your father. And right now, the red strands of DNA are being added up. So these are the maternal strands of DNA, and uh, by now you must have noticed that how there are 23 maternal strands of DNA in red plus the 23 paternal DNA strands in the black. And that's how we know that there are 46 strands of DNA in this cell. This is an example, as I told earlier, that this is an example of human diploid cell. So we move into the S stage of interphase, where we notice what will happen in the chromatin is copied and duplicated. So the 46 strands actually duplicates to form 92 strands. So well, now we go ahead and enter into meiosis, which is meiosis number one. And there is meiosis number one and there is meiosis number two. So overall there are two rounds of cell division in meiosis. So let's start with the prophase number one of meiosis number one. So in the very first step, the nucleus will dissolve. So what happens next is the chromatin will coil into chromosomes, and by now you must have seen that I have numbered them. If you look closely, that there are two chromosome 10s, one in black, one in red, and there are two chromosomes of 15, one in black and one in red, and there is two of each chromosome number, one in black and one in red. And during the process of synapses, these chromosomes pair, these homologous chromosomes will pair with one another. So notice what's happening. Here are two chromosomes, 7s are drawn next to each other. The two chromosome 5s are also grouped next to each other. The two chromosomes of 20s are also grouped next to each other. And these form grouping of what are actually called as tetrad. There's one tetrad of made up of two chromosomes 2s, and there's another tetrad made up of two chromosomes of 5s. There is again another tetrad you can see made up of two chromosomes of 7, and there is another tetrad made up of two chromosomes of 18. And so overall there are 23 tetrads, and I have highlighted just four of them. So there's a question that why does this happen? Why does synopsis occur? Well, synopsis will set up what's called crossing over. And if we focus on the tetrad number 1, for example, so what actually happen in during crossing over is that the enzyme will actually break off portion of the maternal and paternal chromosome and then rearrange them. So if you notice, you have a mostly black parental chromosome with a small portion of red maternal chromosome of DNA on it, and then same with the red chromosome. The red chromosome is made up of mostly red maternal DNA and genes, but there is a small piece of the black paternal DNA in genes attached to it. So this creates a genetic diversity, and this genetic diversity is created in all of the tetrads. And I have highlighted just a few among them, but this phenomena is happening with other tetrads as well. So now have a look at the metaphase 1. What happens in metaphase 1? The tetrads will align along the equator of the cell. You might know that there are threads of proteins called spindle fibers that will attach themselves to the chromosomes, and the spindle fiber will pull the tetrads to the equator, along the equator, or to the middle line of the cell. So now have a look at the anaphase 1. Anaphase number 1, the spindle fiber will pull the chromosome apart, splitting the tetrad. One chromosome of the tetrad was pulled to the left, and the other chromosome of the tetrad was pulled to the right. So, as we move on to the telophase number 1, this cytokinesis will divide this one cell into two cells, and sometimes the nucleus will reform. Now, just for the sake of this tutorial on this animation, I showed the nucleus reforming. Sometime it does, but sometimes it does not. But here, meiosis 1 will ends up. So now let's have a look at what will be happening in the meiosis 2. In meiosis 2, first there will be prophase 2. In prophase 2, if there is a nucleus, then first it will be simply dissolves. So as we move into metaphase number 2, the chromosomes are once again going to be aligned along the equator, or you can say the middle line of the cell. A difference being observed on how it's not the tetrads anymore. The tetrads were separated during anaphase number 1, if you remember. So now it's just the individuals home that are aligned along the equator of the cell. So now have a look at anaphase 2. In anaphase 2, the chromatids are going to be pulled to opposite ends of the cell. One chromatid of a chromosome was pulled upward, while one chromatid of the other chromosome was pulled downward by the spindle fibers. In telophase 2, uh, the cytokinesis will divide the cell into four cells. The chromatids will uncoil into the loose linear strands of the DNA, which are known as chromatin. The nucleus will be reformed. And so when we are done here, notice that we have four genetically unique haploid cells. They are genetically unique because if you remember, they experienced crossing over earlier, and they are haploid. Now, these cells that are created only have 23 strands of DNA, but the one cell we had at the very beginning, which had 46 strands, but this cell only got 23. Same with the cell on the top right, same with the cell on the top left, and same with the bottom left, and same with the bottom right. So this is how the meiosis reduces the chromosome number into half and create haploid cell. So this is how whole cell division, mitosis and meiosis takes place.